Hello, welcome to biology lesson 5.01 over chromosomes. So in this lesson, we're going to be talking about chromosomes. Often chromosomes are considered to be an organelle. So this will be about the fifth organelle we've talked about. So far, we've talked about the cell membrane. We've talked about microfilaments, and we've talked about chloroplasts, mitochondria. So now we're going to be talking about chromosomes. Well, let's start off. Let's take a piece of this. Hopefully you know what this is. Can you tell me? So we've got DNA, and let's kind of expand that. Now, if I untwist this, right now this is in the double helix form. If I untwist this, remember it looks like a ladder. And we've got, I'm going to do. And we have 4 different nucleotides. So the DNA is a polymer, and we have monomers. So let's go back over the monomers of DNA. I have, let's put an orange one in here. We'll put a couple orange ones. Adenine. Let me color these in. In green. I have thymine. And in pink, let's do a couple of pink here. I have cytosine. And let's do purple. I have guanine. And we said you can remember how these pair by using the mnemonic @CG. So adenine and thymine always pair, and cytosine and guanine always pair. In the middle, I have a bond that attaches the nucleotides together. So in the middle here, the bond that attaches two nucleotides together is called a hydrogen bond. And the bond that attaches them along the sides is called a covalent bond. Covalent bond. covalent. We're going to find out that the hydrogen bonds are weaker. That's why we draw them with a dashed line, and we draw the covalent bonds with a solid line. And when it goes into its normal form, it curls up into this double helix. Now, it might surprise you to find out that most of the time during the life of a cell, these aren't present. In fact, these are only present at short periods of time when the cell's getting ready to divide. So the wound up form of nucleic acids as in DNA, in this case DNA, is called a chromosome. And you might look at that picture and go, oh, cool, I've got 2 chromosomes there. Actually, you don't, you've only got 1, but we'll get to that. So down here, the loose DNA, so here it's loose or unwound, is called chromatin. Chromatin. I'm going to hit focus. I think this is a little bit out of focus. That help? Maybe. So how do we get from chromatin to chromosomes? Well, we have a protein that helps us. And those proteins, I'm going to color in brown, are here. Those proteins are called histones. And it's a lot like what you would do. So when you go to the store and you buy a sewing thread, it doesn't come loose in a package. It's usually on a wooden spool. And the thread gets wrapped around the spool, and that helps us wind up the thread. It's exactly what histones do. They are like the spools for thread. So the DNA will wrap around the histones, and then the histones begin to kind of wrap around each other. And we keep getting more and more wound up until we form these structures. I'm going to be doing a lot of color coding of chromosomes, and I'm going to take this chromosome and I'm going to color both sides in the same color, dark blue. Which doesn't really show up here, so I'm going to redraw it over here. And I'm going to connect them in the middle with a black circle. Before a cell can divide, when a cell divides, the offspring we call a daughter cell. So let's just throw that term in here. Start getting some of our terms going. And I'm going to be writing over here just because I know I'm going to run out of space. So when a cell divides, in mitosis, so we're talking about mitosis, we have two identical daughter cells. Well, in order to be identical, that means that each daughter cell has to have a set of the genetic information. And in order to do that, the genetic information has to copy itself. The reason why they're showing this as a copied chromosome is because this happens right before mitosis takes place in a cell. So what we have here are two copies of the same chromosome. And when we have copies, we call those chromatids. Chromatids. And chromatids are attached. They're attached by, let me change back to red so you can see my arrow. By a protein disc. So this represents a protein disc called a centromere. A centromere is a protein disc that attaches chromatids together. Okay, so I'm going to bring this next page on, but before I do, let's just make sure. So check this out. One of the things that students talk about is that one of the reasons this chapter can be challenging, look at the similarity. We have chromatin, we've got chromosome, we've got chromatid, and we've got centromere. Those all sound really alike. You really need to get a handle on your vocabulary pretty early on in the chapter. So I'm going to introduce on the next page something called a karyotype. A karyotype is a picture of an organism's chromosomes. And the reason we're going to look at this is what I want to do in the second half of the video is I want to help you understand what the difference is between a chromosome and a chromatid. All right, so let me pull that other sheet in. So we have two karyotypes, and we're going to come back to this page in the next lesson. But for this one, I just want to take a look at the difference between these two karyotypes. So these could be the exact same karyotype, but let's do some color coding with me. Now I'm going to do a light blue, and dark blue. So for karyotype number 1, and right now we're going to ignore how many total there are. I only want to look at this first set of chromosomes. So let's do this first one, dark blue. And let's do the second one, light blue. Shoot, that didn't show up very well at all. Well, it'll show up better when I redraw it up here. So let's redraw it. So there I redrew. So this first one is this color and the second one is this color. We call this a homologous pair. So if I look this down here, this is saying that this is the first pair. But this is chromosome number 1 and this is chromosome number 2. So these are not the same chromosome. They are different chromosomes. We call these, let me stick with blue, a homologous pair. A homologous pair are 2 chromosomes. And for now, we're just going to say that are similar, but not identical. Again, the whole next lesson is over homologous pairs. So the reason I picked dark blue and light blue is to show that they're similar, but they're not identical. So this is my first pair, chromosome number 1, chromosome number 2. Now over here, I'm going to color code. I have the same karyotype. This is a human karyotype. Let's get this in dark blue. And let's do this one in light blue, which didn't show up, but I'll redraw it again. So now I have this. And I have this. I'm going to attach these by centromeres in the center. Okay, so here's the first question I have for you. How many chromosomes do I have? Well, the answer is still 2, but they're copied. So I still have 2 chromosomes here. But I have 4 chromatids. These are similar. This pair is similar to this pair, but they're not exact copies. Let's go with a chromatid. So let's go here. So if I go 1, 2, 3, 4 chromatids, but I have chromosome 1 and chromosome 2. So I have 2 chromosomes, but I have 4 chromatids. That's why it's important to understand the difference between a chromosome and a chromatid. Exact copies are chromatids. And these are exact copies and these are exact copies. These are similar. Similar is going to be a homologous pair. And again, next lesson is over homologous pairs and why we say they're similar. So our takeaways here. And by the way, again, we'll come back to this page because we're going to talk about the fact that humans have 23 homologous pairs or 46 total. We're going to talk about this last one being special. So, but before we do that, let's go back. So our takeaways, we reviewed the structure of DNA. So we looked at the adenine, thymine, cytosine, guanine, selective bonding, and the hydrogen covalent bonds. We looked at how DNA coils up into chromosomes, how chromatin coils up into chromosomes. When it's loose, it's chromatin. It wraps around these histones, it winds and winds and winds, and we end up with chromosomes. What we have here is a duplicated chromosome. So we have 2 chromatids attached by a centromere disc in the middle. And then we looked at a karyotype to see that when we talk about homologous pairs and when we talk about copies, we have similar and we have exact. So these 2 are exact copies. These 2 are exact copies. And when they're exact copies, we call them chromatids. But when we look at this, we don't say we have 4 chromosomes. We say we have 2 chromosomes. And that's similar to if you have a copy of your report and you make a copy for 3 different teachers. You might have 3 copies of page 1, but page 1 is still just page 1 and page 2. So you have a 3-page paper. You have multiple copies of those pages. So you can think of that in the same way. All right, that gets us going on chromosomes, and we'll see you in the next lesson.